Hello, my name is April Rose. I'm a medical oncologist and clinician scientist working at the Jewish General Hospital. And I presented at ASCO a poster describing our phase 2 investigator-initiated BEAVER trial. So the BEAVER trial was really looking at patients who have advanced cancer that is driven by a BRAF mutation that is called a non-V600E BRAF mutation. So we know that BRAF mutations are common in cancer, and the most classic BRAF mutations are V600 mutations. And we have targeted therapies that work for cancers that have V600 mutations. However, about 30% of the cancers that have a BRAF mutation are non-V600 BRAF mutations, and we don't have established therapies that work for these cancers. So we had done some preclinical research and showed that the same BRAF and MEK inhibitors that we use in V600 BRAF mutant cancers were effective in non-V600 BRAF mutant cancers. And so we designed this trial to investigate the efficacy of Binimetinib, a MEK inhibitor, and Encorafenib, a BRAF inhibitor, in cancers that have non-V600E BRAF mutations. So this was a Simon two-stage trial that we enrolled 23 patients, and the primary objective was to evaluate the objective response rate. We also looked at progression-free survival, overall survival, and exploratory endpoints. So we enrolled patients who had many different types of cancer. About a quarter of them had melanoma, a quarter colorectal, and a quarter pancreaticobiliary cancers, with many other cancers represented as well. And we found that the toxicity profile was very similar to what has been previously reported in other studies. So predominantly low-grade GI toxicities is what we observed. The response rate was 13%. We had 3 patients who had a partial response out of 23 patients enrolled. So this was a lower response rate than we were hoping for a positive study. However, we did see some signals of increased efficacy in different cancer types, especially in melanoma, where there were 2 patients out of 6 who had responses. And then we did a bunch of exploratory studies looking at the genomic profile of these tumors and found that certain biomarkers such as TP53 mutations or altered cell signaling genes were associated with non-response. And we were able to develop patient-derived xenografts from the patients enrolled in these tumors, so we could test additional therapeutic strategies. And what we found is that when we combined SHIP2 inhibitors or CDK4/6 inhibitors with the same BRAF and MEK inhibitors that we used in the study in mice that have these patient-derived xenografts, that we saw much more improved efficacy in the mouse models. So moving forward, we think these triple therapy strategies, which incorporate SHIP2 inhibitors or CDK4/6 inhibitors alongside the BRAF and MEK inhibitors, might be a more effective alternative therapeutic option to explore in this patient population.
Don't forget to like, comment, share, and subscribe to OncoDaily on YouTube. Hit the bell icon to stay updated.